There are three mechanisms that we think about non-dilutive funding. We think about it a lot these days. Small biotech companies think a great deal about non-dilutive funding. First is opportunities like CIRM, mechanisms like CIRM, of which there are almost one. And so this term de-risking that we use to try to explain what we're doing when we get proof of concept in the clinic that something is working, it sounds pretty kind of white bread or vanilla and not very interesting, but actually it's the most exciting moment in drug development when you actually get a sense that what you've been working on is effective in the clinic and is safe. And so de-risking cannot be overstated in terms of its importance. And so organizations like CIRM that actually fund clinical experiments are crucial to the ecosystem. And this is an area that you as investors should keep a close eye on, because what you want is to invest in something that's going to pay off. So that de-risking moment is crucial. The other two mechanisms that I would call out, and have been for years, what are called pre-competitive consortia that involve multiple companies and then typically a government agency like the NIH, which will partner together and put resources into learning something that nobody would take on by themselves. As an example, some cell therapies are going to be applicable in the context of rare diseases. Knowing how those diseases behave in their natural history, meaning without any treatment. So you can understand what to expect if a treatment works. Getting that sort of information can be carried out by these public-private consortia. The other thing that they could do, and I think will be essential for our ecosystem, is to get real-world data about what happens when you give this type of cell therapy for that indication. So actually to establish the database that we're going to need to address the challenges around reimbursement. So those are two areas. The third one, small biotech companies in partnership often with academics can apply for funding for grants, just like an academic investigator. Some of the work that we do around cell therapies is in fact basic science. So we always have to have a potency assay. Is the cell doing what it's supposed to do, at least in a tissue culture dish, to give you confidence it'll work in the human being? One person's potency assay is another person's fundamental T-cell biology. So you can apply for support to generate that type of data. Another type of information that we are going to need, particularly in the autologous cell therapy space, is to understand, why does one person's cell preparation turn into a fantastically effective cell therapy and the other person's not so much? Is it the person? Is it the condition? Is it genetics? Is it other medicines they're taking? These are basic science questions. Grants can be written to support the investigations that will get us those data. And those will be catalytic for you as investors to bring these therapies forward further into the clinic.
Fantastic. I think that's basically in the matrix for evaluating the cell therapy when you're looking at a potential de-risking. Kristen, what do you see for the red flag? You work with a lot of startup companies.
At the end of the day, it comes down to the science. How do you solve the problems that have been identified with the first generation of products? Those are often scientific problems to solve, and so you need to have a credible hypothesis for how you're going to tackle that problem, and then have a technology to do it in credible science to support that hypothesis. What I would say is, it's always hard when you're evaluating early stage companies and animal studies to pick the winners. If you just look at the field of cell and gene therapy and the typically incremental change in the advancement of science, with a lot of first in class and then me too, I'm going to be best in class type approaches. And then you see these quantum leap changes. Quantum leap changes have already occurred with CAR T cells, first in blood cancers. Now they're moving into autoimmune disease. 3 years ago, the very first presentation of a response of a CAR T cell in a lupus patient was presented at the American College of Rheumatology meetings. 3 years later, 30% of all presentations at that conference have to do with cell therapy for autoimmune disease. So as an investor, the question is, do you want to be in a field where there is going to be quantum leap changes? Or do you want to work perhaps on the slightly less risky Me Too, best-in-class angles for the 13 cancer targets that have been targeted for the last 50 years? So yes, there are some red flags, and you have to do your deep scientific diligence to believe that that next generation technology is going to tackle an important problem. But fundamentally, engineering the immune system to treat human disease, not just cancer, autoimmune disease, cancer, maybe neurodegenerative disorders, organ transplant, affords the opportunity for quantum leap change. So if I was an investor, I would invest.